I'm happy to be here. I have to say a few things to start. So one is I'm not I haven't looked I'm new to sea ice, so brand new essentially. So I don't know much about sea ice. Um, my background. So I do have a fluid dynamics background at some point, and I've been interested in microbial communities in the environment and how they interact with their environment, and in particular physical and chemical interactions. And I've looked at situations, fairly extreme environments where those communities really can't get away without um, paying attention to their physical environment. So um, I met Noah, and this seemed like an interesting case of such a thing, and I've got involved in it. So that's one thing. The other thing, this seems to be at a much smaller scale than um, everything else that I've heard so far. And it's not really specific to Ms., although there's some interesting aspects maybe of Ms. here, but I won't really um, talk about them except to mention them where they may be. But I'm extremely interested in anything anyone has to say about it. Okay, so all right, so these are the people I'm working with here. This is their work with Noah, and then a couple of microbiologists here at Temple University. So the thing that struck me was there's this microbial community that's living in the bottom of sea ice quite often. And this seems like a difficult environment because, how do you live in ice, actually? So it's not so much the temperature as how do you transport things in and out? And so there's a couple of pictures. This is, I think, probably a pretty well-known picture. This is a ice core. It's taken out of sea ice with a bunch of green stuff left over. So there's slushy stuff going on here. And this is a diatom that we actually we're using in an experiment that comes from ice. It's remember it's something like 5 to 10 microns long. So smaller than the kilometer scale that we've been thinking about. Okay. So yeah, so I should apologize. I don't know what's familiar and what's not familiar to everybody. If I say something that's too familiar, oops. If I don't, then just stop me and ask me. So before we get going, a few things about sea ice that are important here. So uh, sea ice, well, from my idealized point of view that we'll use here is basically our seawater is salt dissolved in water. The ice itself is essentially pure water, which means that as ice forms, the salt has to be sequestered in brine that gets increasingly salty as more ice forms. And the freezing temperature in that brine will depend on that concentration. And so you get to, depending on the temperature, you get to a balance between the amount of ice and the amount of brine, right? More ice means less brine, but it's saltier. So eventually you get to a equilibrium. So as a consequence, sea ice is not pure ice. There's liquid brine in it, and that's the place where microbes could possibly live. And it's interesting that there's an interesting structure, of course, in the ice, which is colder at the top generally and warmer at the bottom generally. So there's more brine volume traction down at the bottom. So that seems promising for microbes if they can set themselves up down at the bottom. If there's enough brine there, then you might actually be able to generate some flow and communicate with the underlying ocean. Okay, so here's our hypothesis about how these microbes might be working. And just a quick word, I mean, I also don't know much about microbial ecology in the ocean. I've worked in other places, but we're thinking of photosynthesizing organisms essentially, although there's a lot of other stuff there. So they have a lot of energy. They're generally not energy limited because they're getting energy from the sun, but they do need certain nutrients, and they have to get rid of the trash as well. So they need to be able to communicate with the ocean somehow to transport things in and out. So the idea is that the hypothesis is the following. Microbes can produce osmoite, which is, for our purposes, effectively salt, extra salt. So they make the brine saltier, which will actually melt the ice a little bit. So it raises the freezing temperature, local freezing temperature, so that'll melt the ice so that the salt, their brine becomes less dense. So that salt boost increases permeability, and that allows fluid flow. So we also have to worry about how that's driven, but that'll come later. So that's half of it. And then the other thing to keep in mind is that if you produce this salt, this osmoite at a given rate, then to get to an equilibrium state, the transport rate has to match the rate that you're producing things. If you're producing salt faster than the flow is transporting things around, then you'll build up more osmoite, get increase the permeability, more flow. If you're producing salt at a slower rate, then your channels will start to close up because there's not enough salt to replace what's left, what goes from the flow. So it's very easy to regulate in principle. The microbe just says, I need flow at a certain rate to give me nutrients, and I'll produce osmoite at that rate, and it should work. So that's the idea. Okay, so this maybe should have been the first thing we did, but it's actually sort of the last, was to go to the lab. And we took some of these diatoms, which I showed before, or well, our friends over in biology did this. And we just wanted to measure the freezing point with and without these microbes. So had a pretty simple experiment where we took some stuff of some sort and put this in a bath of dry ice and then measured the temperature, and I'll show you what they got. So these are temperature versus time measurements. The first one is with just artificial seawater. So you put this in, track the temperature, it super cools, and that at some point starts to freeze, and it jumps up to roughly -2 or -1.8 degrees, as it should. So that's a type of control. Then we took a solution of, so these guys had a culture. They're growing in their refrigerator at close to freezing of this diatom, and they took some of that and put that in and did the same thing. And so we get the supercooling, and then freezing occurs at roughly a little bit more rather than -3 degrees. So approximately a 50% decrease in the freezing point temperature. So they tried to use a concentration that was something similar to what you might see in ice. Just to check, so on the right, so what they did was they took some of this culture, and then they spun it down in a centrifuge and pulled off the supernatant. So you have this tube, and the stuff, all the heavy stuff gets to the bottom because of the centrifuge, and they pull off the fluid on top, what's called the supernatant, and used that. So, and we got essentially approximately the same. So actually a same depression. So a lot of this is explained not by the organisms themselves or any kind of the heavy stuff that's in there, but what's in the supernatant apparently. Okay, so this result says, at least in this case, so this is far from growing stuff in actual sea ice, but so one has to take it with a grain of salt, so to speak. Got roughly a 50% increase in effective salinity here. So that's significant, right? There is an effect there. Okay, so the third part, I guess, of all this is so you open up the permeability. You still need to drive flow. So we piggybacked on what some other people had been doing for decades now in the lab. Worster and others, show a picture in a second. The idea was that you can get a Rayleigh-Taylor instability, which happens because you have heavier stuff above lighter stuff as you go down through the ice. So the brine is more condensed higher up where it's colder, so it's heavier, it's more dense, I should say, than below, and that can drive an instability. And the key number is this Rayleigh number here, which is roughly speaking, it tells you if at a given location how long it would take to equilibrate just by diffusing versus how long it would take to equilibrate by flow. And so if that Rayleigh number starts to get large and the flow time is relatively short compared to the diffusion time, then you can actually get some convection going. The key, I won't discuss all these parameters, but pi here, capital Pi, is the permeability. So the idea is that the microbes can control that. They can increase that or decrease that as they want, perhaps. Delta rho is the change in density, which is also important. Delta H is the change in height, and some other things going on here. Okay. So here's a picture from one of these papers of this. So this is lab sea ice. It's water with sodium chloride or something in it, just regular salt, I think. And so they start growing ice in the lab, and what they see, so here is the ice, and this is the water below. So the ice starts moving forward, and in fact, so you get this Rayleigh-Taylor instability actually to go because you have denser brine up here, which is more salty, and will start convecting. And these are actually plumes coming out of salty water that's dropping out. Here's a cross section, and these plumes correspond to these things. You get actually they call them chimneys, downward chimneys that go out. And of course, if stuff is going out, the fluid has to go back in, and that's occurring, seeping in elsewhere. Now, so this is fine. This is good. I mean, this produces convective flow, but it stops when it's only temporary, because you need you need to freeze new salty water to get new salt to drive this thing. So once you lose your salt or enough of your salt, this will stop. Okay. So I don't want to talk about the details so much. I don't know exactly who's interested in what in this sort of group. It's easy to talk about things at other times if you want to. But these are essentially the equations we're dealing with. So the old equations are the top two that people were using for explaining the sort of pictures I've been showing. H is enthalpy, or heat, essentially, roughly speaking, and S is salinity. So we're tracking these two. We're adding, in particular, this third equation is important. This is the new salt. We call it osmolite. So all of these equations, they have a transport and diffusion, although some of the diffusion terms can be neglected. And in the osmolite case, there's also which is the important new thing, there's a source term. So B is some measure of the microbes, and proportional to them is some production rate, which can depend on various things. We may, in some cases, I'll show you, we're tracking actually B, and in some cases, we don't have to do that. But there's a similar equation, possibly, for the microbes. And there are other things going on here as well. We need to compute brine volume fraction. This comes from this relation I talked about before between brine concentration and temperature and ice and stuff like that. And then there's the fluid equation. So it seems to be typical to use Darcy's law inside of the ice. So we're fine with that, although we simplify down in a bit. It looks like this, and there's a buoyancy term, which is actually driving the action. Now, one thing that's not talked about so much, as far as I can tell, are boundary conditions or interface conditions between the ice and the ocean below in particular. It's not such an issue, I think, on top between ice and air. But at the lower boundary, especially if we have flow going from ocean to ice and vice versa, it's not that clear what to do there. And that's super important, because that's telling you, well, for example, that's telling you how much heat is getting transported through the system. So we don't know that much about it, and it seems important. It seems likely to me that the underlying fluid dynamics are important. So all the stuff about waves here this is one place where this is actually matters, or could matter. in a couple of ways. One is it could actually drive flow potentially by pressure differences, and it also impacts transport of heat and nutrients. So it could matter. Okay, so this is basically what I've been talking about. If you can actually drive Rayleigh-Taylor instability off of Darcy's law, if the Rayleigh number gets large enough, and that's been done by various people for Worcester and others for a while. Okay, so somebody at some point, these people, I'm not sure I even have all the right references here, but at some point, um, so this kind of process is super complicated, even as it is, but we don't know what the right constitutive laws are for sea ice, for mushy sea ice. So even if we knew, it'd be super complicated to do this, but we don't even know what they should be, and they're probably pretty complicated. So at some point, an idea was introduced, which we will piggyback on, is to replace the flow by a 1D parameterized flow. So we only keep track of stuff going upwards. This is the seep flow from below. And then there's sort of a magical chimney that takes things away when the Rayleigh number indicates there should be instability. So it's a very crude approximation, but it's an interesting one. You could get some interesting results. So we've set up a system, pretty straightforward, central differencing mostly, with a essentially conserved volume type stuff to do this. I'm just showing this in case there's anybody who worries about these things. Nothing fancy there. Okay, so let me show you a couple of models that we've put together for the biology. Actually, this isn't here. So this is just a repeat of what we had before, with the addition of a sea compartment, so we can keep track of microbes in the sea, so we can communicate, have stuff flow out of the ice into the sea and back into it. Okay, so here are the two models. The first one is call it a biofilm model. So that means all that means here is microbes are actually stuck to the ice and they're not moving. So we have a super sophisticated biofilm model, biofilm model version 1, which is just b equals 1. It's constant. So there's stuff stuck to the ice. And then in addition to that, have just a slightly more sophisticated planktonic version where the microbes are not stuck to the ice, they move with the flow. And here we have a sea compartment. Not worrying about limiting nutrient at the moment. It's not really central to this, probably. There's some other things going on. We have activity. We need to inhibit the activity somehow. So if there's effective, if the salinity gets too high, then we'll do that, et cetera, et cetera. Okay, so here's an example solution. And what I'm plotting here, so plotting the velocity, the upwards velocity. So we have a velocity here that's moving things up, or it could be 0 in upper regions of the ice. And so that's plotted here versus depth. So up here, there's no upward velocity at all. Time is this way. And you can see this is the biofilm case, and we actually get, that's kind of interesting, it's like a periodic geyser, sort of. So this is stuff that's moving out, and I'm not showing the stuff that's coming out. Moving up, not showing the stuff that's coming out. But so what's happening is, so this is where the microbes are stuck to the ice, and they start producing osmolyte, and it builds up, and at some point we cross the threshold, the Rayleigh-Taylor threshold that allows instability to occur, and then suddenly the whole thing sort of empties itself out, and the process starts again. It's quite robust, by the way. I mean, as it should be. If the microbes are producing things at a certain rate, as I said before, the flow has to, on average, transport them away at that rate. On the right is a planktonic case. So this is where the microbes are not stuck to the ice. They just move, and you can hardly see it, but Again, so there's a much smaller layer. This one is basically, this is a 1 meter ice sheet, and it's basically 10 centimeters. There's some extra stuff going on above, but that's another story. So here's, I think, 3 or 4 centimeters thick. And it takes a little while to settle in, but you do get something going. In the planktonic case, the issue is that this flow can actually wash out the bugs. And that's essentially why. And if they don't actually produce osmite fast enough, the whole thing gets washed out, and you don't get anything going. In this case, so it's less robust here, and the layer is a lot less thick. Okay, so I thought I have little time and just throw, and there are other things to talk about here, but this one is kind of interesting. So I'm looking here at the temperature change from time 0 to later time. So this is the difference in temperature at this time, about 1,000 hours versus 0 hours. And I should have said, so for initial conditions, what we do is we run this code without any of the biology until it settles down to approximately steady state, steady profile. And that we use that at t equals 0, we introduce suddenly microbes, and it takes a little while, but they set up this periodic state. And now, so we're looking at the difference in temperature. And so we have this flow coming in from below, and as you'd expect, that actually increases the temperature because we're bringing in warm water rather. But in fact, the net transport up above, the temperature actually decreases. And then, in fact, the net transport of heat, somewhat surprisingly to me anyways, decreases because of this fact. And the reason was that, yeah, so you get transport of warmer water down here. But the extra brine, so the brine is much less conductive of heat as compared to ice. So all the extra brine actually makes the bulk properties, transport properties, such that you get less transport of heat. So this, so we see that here, and this could easily be an artifact of the model. It's the microbes shouldn't really be producing osmoid up here, and they are. But just to note that it's actually a little bit subtle. In the planktonic case, where there's not much flow, in fact, the temperature decreases everywhere because of this effect. Okay, so let me finish then with some final remarks. We see microbial communities in the bottom of sea ice, which to me was surprising. This is not Ms. specific, I think, but in a lot of places, because it should be hard to live there for reasons I discussed. And the first thing that comes to mind for me is transport. How do they get their nutrients, and how do they get rid of byproducts? So and the hypothesis is that microbial activity can easily help with that. But so I didn't talk about it this much, although I guess it did a little bit. So there may be some effects on sea ice material properties in all of this, including mechanical ones, right? If you're opening up pores and things like that. They're based on our extremely crude model. I don't see any reason to claim that they're going to be large, but I can't really say that they wouldn't be. And I would like to propose a question. Maybe people out there somewhere have thought about this. How do the underlying ocean dynamics interact with flow inside of the ice itself? Do they help drive things? Okay, so I'll stop there and take questions, if there are any.
Thank you, Isaac. Any questions? Yes, Pat.
Hi, Isaac. Yeah, thank you very much. Just to ask one question, when you meant osmolyte, you mean dimethyl sulfonyl or DMSP, or you mean also other?
Now, well, we don't, for purposes of the model, we don't care what it is, but it could certainly be that. Or it could be almost anything, right? There's lots of things that possibly could do that, and we saw that in our temperature measurements that there's certainly something in there, and it could be DMSP or maybe something else. I don't know.
Yep. So that's mean for the next step, it could be nice to include gas in the model, or what's your plan for the next step for this? work. Yeah.
No, I'm curious why are you interested in gas? Why do you think that would be a thing to do?
Oh, because if it's a DMSP, then it could link with DMS, those cycle as well. Would
Yeah,
that be interesting to you to have in that yeah.
for sure. But I was under the impression that it would still be dissolved. Eventually, I mean, it's volatile, so it could leave. But I'm not aware that gas phase is an issue here. But like I say, I don't
Okay.
know that much.
Yep. Okay. Yep. Thank you very much, Isaac. Yep.
Thank you. Marcelo.
Hi, Isaac. It's a great presentation. Thank you very much. Enjoyed it a lot, and particularly all the details that you gave us. So we are actually doing experiments similar with the hellish show cell in 2D by looking at streamers and all the rest. And we are starting to think about biofilm and including all of them. I wonder if you're familiar with the work done by Ruckertz. I think Arnaud Ruckertz has been published a few years ago about the paradox observed in productive CIs, where we see an increase of nutrients when you have high biomass, high phytoplankton biomass. And this paradox is actually being found anywhere, not just in the bottom of the ice, but in Antarctic sea ice, also in the middle of that.
Oh,
So
okay.
I think all of what you're actually telling me is that the biofilm is essential to not flush out all of that. So depending on, but we do see also in the middle where you have shown this increased, lower temperature, because your brine should be less permeable, less communicative. So I wonder how, because that would move everything kind of closer to the bottom of the ice without not allowing anything in between. Do you think there could be something else at play? I'll share with you this link so you can look at
Yeah.
that.
Yeah, I mean, I've heard of that, and I think I've seen that paper. So I mean, it's entirely consistent. More nutrients is entirely consistent and in fact necessary with transport in the bottom. I wasn't aware of that happening in the middle of the ice. And for our particular model, I wouldn't really trust what's happening in the middle of ice so much because we don't really care about that. And it's likely that the microbes would be fairly inhibited there, and we're not really doing so much of that there. So it's just as long as they're not able to actually produce channels that open into the sea, then it doesn't matter so much in our model. But yeah, I mean, I don't have anything to say, and it's a surprise to me actually that there would be more nutrients in the middle of the ice. So yeah, can't answer that.
Will you be publishing this one soon? Because we are really interested.
Yeah, we have a submission at the moment.
Okay.
So
Excellent.
I'm happy to send a preprint if you want to see it.